Okay.
Okay, let me just move my screen so I can stand. Usually it's easier for my daughter. Sorry about the light contrast. I think you're gonna see me. Maybe once my husband comes back, we can take a short break and I give him the kit, and then I'll move a bit so you can see the better contrast. Now you're gonna see the sunlight and me against the sunlight, but
Sure.
you can still see me and hear me.
Yeah.
You see this the right screen for the slides, right?
Exactly. Everything is totally fine.
But I can't hear you. Trent, can you say something?
Yes, I'm saying something.
Are you unmuted? I definitely can't hear you.
Can you hear me?
Okay.
Yeah.
Okay, now it's all good. Okay.
Okay.
Great. So the second set of lectures that I will give today go into using some of the methods and the techniques that I covered in the last lectures, namely on how to model synaptic plasticity, activity-dependent synaptic plasticity, and I will talk about them in the context of my research topic, and that is development of neural circuits. So I'm very interested in how young brains basically go from being, quickly and imprecisely connected, as in the brain of this baby who happens to be my nephew or my daughter here, to being strongly and precisely connected, obviously non-randomly connected into, as we transition and mature into adulthood. Mm-hmm. Now we know that this is a complex process, that it involves a large number of mechanisms. Broadly speaking, we can divide them into two classes. So one class of mechanisms that I like to call activity independent. So these are ones that depend on interactions between sets of genes and molecules. And of course, the ones that I'm very interested in studying, so I just want to show you the schematic that kind of explains what I mean by these activity independent mechanisms. For example, in the context of the developing visual system, we know that there's these molecules called Fs and effrins, and they basically, there's sort of several of them, and they're distributed in this sort of 2D plane. If you think about the, vision, and then how when we see, even though the world is three-dimensional, it's kind of mapped on this two-dimensional layer, which is our retina. And so there's these molecules that basically that are distributed in gradients and provide a very coarse and rough map of where axons are supposed to go. So in this case, we have a schematic from the retina to the superior colliculus. So this is where the projections go. And so in the retina, we have the nasotemporal, the dorsoventral direction. And so we have the distribution of these different molecules, for example, the FBs that go from low to high gradient of concentration in the dorsoventral direction. And then we have these two effrin A5 and FA5/A6 that are basically opposite distributed, meaning the high gradient for one of them in the nasal plane is actually low in the temporal plane, and vice versa for the other molecule. And the interesting thing is that then they get matched to corresponding receptors in the superior colliculus along the lateral medial direction and rostrocaudal direction. So these activity independent mechanisms are super important. They are kind of thought to act first. They're thought to be there way before the onset of any kind of activity. And they're thought to really set up this coarse map of connections. So really telling axons in the retina approximately where to go. I say coarse map because they're determined by these, by sort of these axons have to sense, these growth cones have to sense kind of location, the amount of gradient in a particular location. This is not very precise. And so the maps that they form are not super precise. And so it is known then this second set of mechanisms, the activity dependent mechanisms, the ones that I'm interested in, actually play then the next most important role to refine this connectivity that is initially set up by these activity independent mechanisms. And as we, normally, when we normally think about activity, we normally think about sensory evoked activity. So we're usually studying the sensory cortex as an area where we're trying to see how it develops. And here, in the context of vision, of course, we talk about input that comes from the eyes, so visual input. There's, of course, auditory input, olfactory input. So basically all of the senses generate this sensory evoked activity that we know activates these activity-dependent mechanisms such as spike timing-dependent plasticity, Hebbian mechanisms, and so on, to then refine these connections. But in the context of development, we now know that many organisms, including humans, right after their first sort of put together, let's say in the case of rodents, for example, even after they're born, many of the sensory organs are immature. In the human, the organs are still less mature when they're born, but there's quite a bit of development that happens already in the womb. So you may then wonder, if these sensory organs are immature, can they actually receive properly, as in the adult organism, can they receive properly this sensory stimuli to drive these refinements? And the answer is sort of yes and no. And the answer is no, because they're not very mature. So, but yet activity is still somehow there. So let me first try to convince you how this activity is important for refinements. And then I will tell you where this activity comes from in these immature organisms when these sensory organs are basically very young and cannot really transduce the sensory information as well as in the adult. So I want to show you this video. This is from a paper in 2013 to try to motivate for you that activity is indeed important, that it's not just all about genetics and molecular interactions. So in this case, there were these animals that had a genetic mutation that prevented them basically from, I don't know, they were viable, but so they could live, but they had a shortened lifespan and they had severe deficits in behavior. And so, but in this case, if they let the animals, rather than live in their cages, get exposed to all sorts of sensory behaviors and experiences in these so-called enriched environments, they were able to find that despite the very different genetic background of these animals, they could still, develop into perfectly normal animals that had a range of behaviors like their normal counterparts. So this is just to say that, in this case, the enhanced sensory activity was sufficient to overcome the genetic deficits in these animals. So activity is indeed important for these refinements. And as I already started explaining in the previous slide, in these very young animals, many of the sensory organs are immature. So rather than activity coming in from the sensory periphery, it's actually spontaneously generated by many of the developing circuits. And so I've spent, a lot of my years as a PhD student and now in my own group to investigate these patterns of spontaneous activity and to try to demonstrate that they are indeed non-random and they're important for driving mechanisms like Hebbian plasticity and STDP to refine connections that are initially set up by these interactions between Fs and efferents. So let me give you an example of what this spontaneous activity looks like. And this is actually first an example from a human. This is activity recorded in a 30-week post-conceptional age infant, and it's recorded in the somatosensory cortex. And in parallel to this activity, this is EEG, in parallel to this activity, the movement that this baby made was also recorded. So what you can see from the movement trajectories is that they're basically flat. So this baby is basically not moving. However, if one looks at the activity in the somatosensory cortex, you see these very stereotypical elements of activity. For example, these regions colored in gray, the gray boxes, that are called delta brush oscillations. So these are fairly typical for these developing babies. And because they basically don't correlate to any patterns of movement, this sort of demonstrates that they are indeed spontaneous. So they're not evoked by any kind of sensory stimuli. The spontaneous activity is not just seen in sensory areas, even though this is going to be the focus of my discussion, of my presentation today. It is also seen in the development of motor areas. And so let me give you one example of what this looks like. For example, here I'm going to use even a very different organism, so not a mammal, but Drosophila. So this is the fruit fly that you may have seen on, if you forgot apples or bananas rotting in your kitchen. So these are studied by my collaborator, Jimena Berni. So this is the Drosophila. It's quite small, it's less than 1 millimeter long. They can go through different stages before they pupate and become flies. This is recorded from the bottom. Actually, Chuen knows this. I built models of this when I was a PhD student and postdoc. So what you can see here is these beautiful patterns of crawling that the animal generates by muscle contraction. So the left and the right side of the body, that in this case is symmetric when the animal goes forward. And so I hope you're convinced that the pattern of muscle contractions are very stereotypical, very, very regular. That's what enables the animal to just go forward and sometimes turn by generating left-right asymmetries. However, we can look at this organism before hatching, so while it's still in the eggshell, so before it becomes a larva in the embryonic stages, and this is what it looks like. So this animal cannot crawl, but I hope you see that it's still very much capable of generating these spontaneous muscle contractions. And through various experiments done here by Sarah Crisp, now more than 10 years ago, the authors were able to find actually that a lot of these muscle contractions are indeed neurally driven. So if you block synaptic transmission, you can completely eliminate them, suggesting that they are indeed driven by spontaneous activity, just like in the sensory cortices in mammalian organisms, as I showed you before.
This is before L1 stage.
Yeah, sorry?
This is before L1 stage? This is during the embryo?
Yeah, this is before hatching. And it's really actually interesting. There was a Journal of Neuroscience paper 2 years later in 2010. I really wish they followed up on this, but they actually identified a critical period of plasticity where the spontaneous activity that they see in the embryo just before hatching seems to play a super important role, just like in mammals. So you'll see that in the second part of the talk today, I'm going to focus on basically the period in mice after eye opening, that's known as the critical period of plasticity, because if you perturb activity in this time, and you'll see an example of that, the brain is extremely plastic. So these are like the classical experiments by Hubel and Wiesel. And if you do the same experiment later in adulthood, you don't see the same plasticity. So that's why this is classically termed as the critical period of plasticity. And so also in these Drosophila larvae, if they do a certain set of manipulations and they block, I think, this activity or synaptic transmission in a certain window just before hatching, they also see that they can completely delay hatching or completely prohibit hatching. So there's very curiously this critical period of plasticity also in the motor system of these animals. Very paradoxical. I mean, I know Chuen and others study other motor programs in other organisms like C. elegans, and classically the idea is activity plays no role, everything is genetically predetermined, end of story. But that's actually not the case, at least not in Drosophila. Mm-hmm. I don't know if C. elegans has spontaneous activity, if you can follow it before it becomes...
Well, in C. elegans, it starts to sort of move around within the egg, hatch a little bit, but at L1 stage, they already start to move pretty much like the young adult, with a completely not, with half of the motor neurons that you have in the adult motor circuit. Yeah. Mm-hmm. Mm-hmm. So, but it's also very interesting. It's a very different circuit, but can do pretty much very similar things.
Sure. Yeah. Yeah, so I just give this as an example just to say that, you know, this activity-dependent refinement that I will talk about, driven by spontaneous activity, we don't think it's just unique to, you know, mammals and sensory systems. So that's why I like to give this example. But what I will spend more time talking about are activity patterns, in this case generated spontaneously before the maturation of the sensory organs, in the sensory system, I will use mostly the visual system as an example of rodents. So here in rodents, in mice and rats, eye opening is 15 days, so about 2 weeks after the animal is born. And in this period of 2 weeks, even though the eyes are closed, this is denoted here in the schematic, the brain is not silent, it's experiencing these patterns of spontaneous activity. And if we talk about the visual system, these patterns of spontaneous activity are literally all around the visual system. So they are in the eyes, so in the retina, which is the first circuit that processes vision, and this is how they're manifested in the eye. They propagate in the form of what I call retinal waves, or others also call retinal waves. They were defined and first found by Marcus Meister and Rachel Wong in Carla Schetz's lab in the 90s. They were first identified actually in vivo by Rachel Wong, but also then characterized by Marla Feller and Marcus Meister in vitro. So they usually take out the retina in a dish. In this case, they put calcium. So the calcium indicates fluctuations in the activity. And I hope you agree with me that this is definitely not random patterns, right? This is very beautifully spatiotemporally patterned, and it really propagates like waves. So many people have studied these waves, and still Marla Feller publishes to this day very nice papers identifying more interesting aspects of these waves. They basically change as a function of development, different circuits underlie their generation. And in what I'm interested in is actually how these patterns drive refinement of connections downstream. So remember in one of the earlier slides where I motivated these Fs and ephrins, I showed you how connections from the retina grow to the colliculus, but also to the visual area of the thalamus known as the dorsolateral geniculate nucleus, and then going to the cortex. So we're usually looking at the cortex. So let's look at what this activity looks like in the cortex. One can do similar type of calcium imaging. So you put either genetically encoded calcium indicator, or you can put a dye and then basically load the cells with calcium. And again, here, this influx of color that we see basically denotes propagation of activity that is generated by, generated basically spontaneously. I should just point out, let me play the two videos again, that we're talking about very different spatial scales here. So at the top, the side of the rectangle is about 0.5 millimeter. So this is actually about 1/3 or 1/4 of the entire retina. But at the bottom here, we see a very small window in the cortex. So here the scale is several hundred microns. So at the bottom here, the individual blips are indeed cells, and at the top it's much more difficult, so you cannot really discern single cells. So we just have slightly different spatial scales. But the other thing I want you to notice is that although this retinal activity propagates like these waves, in the cortex, we think that the activity is not exactly like waves. It's kind of, we think it's more modular. So we think that there's sort of areas that kind of go up and down in more of a all or none manner. And, you know, we have people looking at this activity. In fact, I think one of your students, who I think is in the audience, Joshi Liu, is actually doing her master thesis in my group, and is looking at these patterns of spontaneous activity and their role in refinements in healthy and also in diseased brains like Fragile X animals. So we really believe that this spontaneous activity is non-random, and we and others have demonstrated this. And we're interested in using these mechanisms that I talked about in the last lecture, these activity-dependent plasticity mechanisms like Hebbian correlation and covariance rules and STDP, and how they refine connectivity in these developing brains. So what I want to show you today are two examples of some recent work that we have done that address these ideas in the context of development. And I will do this kind of in two different dichotomies. So the first dichotomy is I will address two different aspects of development. I will first address the stage of very early development, right before eye opening, when this spontaneous activity is very prominent. And I will ask exactly how these spontaneous activity patterns refine circuit connectivity. And then I will ask, once we know that these circuits are set up by spontaneous activity, I will then ask how these circuits are then maintained by visually sensory evoked activity after the onset of sensory experience, so after eye opening. So in the second part, we're going to look at basically a functioning network. And then we're going to apply a perturbation mimicking an experimental perturbation of sensory experience. And then we're going to study activity-dependent mechanisms that try to maintain circuit function and circuit organization in the presence of the perturbation to the visual experience that was applied. So this is the first dichotomy. We're going to look at these two different developmental time points, very early and after eye opening. And the second dichotomy is I'm going to show you mathematical models of how we do this at two different scales, spatial scales in this case. So we're all interested in studying the brain and how the brain works. So this is just one image taken from the brain atlas, mouse brain atlas. And so what we see here are, of course, neurons that are interconnected into networks. And the way we can study these circuits is either by focusing on sort of networks of neurons. So this is the scale of microcircuits, and this will be the focus of the second work that I will describe today. So we will be looking at models of single neurons where we just model each neuron as a point neuron. So you're going to be describing the activity of the neuron through the memory potential and the spikes. And we're just going to be interested in how synaptic connections modeled by these weights that change in time are influenced by these patterns of activity. But in the first part of the talk, I want to take this, still discuss sort of network interactions, but at the so-called subcellular level. So rather than looking at networks of neurons, where the neurons are just point neurons, I'm going to study interactions within the framework of a single neuron. So the interactions are now not going to be between neurons, but they're going to be between individual synaptic inputs on a given neuron. So if we zoom in, rather than looking and modeling the neuron as just a point neuron, just focusing on the cell body of the neuron, the soma, we're now going to take into account the entire dendritic tree structure of the cell. And we're going to specifically investigate these synaptic inputs, which are the points of contact of other neurons. So the input that other neurons provide to this neuron, indicated here with the red circles. And we're going to study just how random or non-random these synaptic inputs are distributed on the dendritic tree of the neuron. So one dichotomy is different developmental time points before and after eye opening, and the second dichotomy is spatial scales. So we're first going to start looking at early development and organization of these synaptic inputs at the level of the single cell. And in the second part of the talk, we're going to move into later development, and we're going to be studying networks of neurons where we only care about basically the cell bodies, and we're going to represent all of this. We're going to effectively average all of these synaptic inputs that the neuron receives and just describe them with a single synaptic connection, kind of like what I was showing you in the lecture on Monday. So I hope that's clear. So this is it for the introduction part. I'm now going to dive into the research topic. And if you excuse me, I'm just going to take like a 30-second, this is not a real break. I'm just going to take a 30-second break to just leave my daughter with my partner. So just give me a moment. I'll be right back.
I think it's quite clear so far, right? If any questions from the students, you can write on chat room.
Okay, any questions before I move into the first topic? No questions. Okay.
I think
so.
As
I said, no. Say again.
I think it's very clear. Yeah.
Okay, perfect. Okay, so the first part is very early development, and we're going to be looking at organization of these synaptic inputs on the dendritic trees of single neurons. So what we have here is actually a pyramidal neuron, excitatory pyramidal neuron, from the visual cortex, but equally from the hippocampus. So let me just actually move my computer. So we're going to be looking at the organization of these synaptic inputs that we know are established developmentally. So these were experiments that were done actually not long ago, literally only 10 years ago, in the lab of my collaborator, Christian Lohmann. What they did is they did 2-photon calcium imaging of individual synaptic inputs in these developing neurons. So here's an example of what they did. They took individual portions of these dendrites and just looked at the activity at synaptic loci, so synaptic inputs. So here are 2 synaptic inputs shown in each of the 2 figures. And then they measured the amount of coactivity or correlation as a function of distance between the synaptic inputs. And so they discovered a relationship that's very beautifully demonstrated in this figure here. Namely, the amount of coactivity or correlation basically decays as a function of distance, and actually quite rapidly. So this suggests that synaptic inputs that are nearby each other, so within 15, 20 microns of each other, tend to be highly correlated. So they tend to show high amounts of coactivity, and this amount of coactivity decays as the inputs get far away from each other. They identified this in very early development, around postnatal day 7 or 8, so right at the transition from the first to the second postnatal week. And they found that this is driven exactly by spontaneous activity, because when they block activity, they found that this particular relationship disappeared. So it's something that is set up because of spontaneous activity. They call this property synaptic clustering because it just tells us that these synaptic inputs tend to be clustered according to how active they are. So it's functional synaptic clustering, functional because we don't really know what kinds of stimuli these inputs will respond to. This is all spontaneous activity before the onset of sensory experience. So that's why we call it functional, because it's measured by the amount of activity. And we call it clustering because it means that inputs that are coactive together tend to be near each other. So spontaneous activity can clearly wire up these circuits at this subcellular precision. And so we wanted to understand how this happens. So this is work done by my great PhD student, Jan Kirchner, who initially set to do this, to basically take a portion of dendrite, distribute synaptic inputs. So this is our modeling perspective of the circuit, and distribute these synaptic inputs in a random fashion throughout the dendrite. And then what we wanted to do is basically come up with a model that tells us how these synaptic inputs will reorganize, that will hopefully give us these kinds of, synaptic clustering relationships. So how do we set up the model? Well, we needed sort of several ingredients. First, we need some kind of input to drive these synaptic inputs. Now, I told you this is early development, so it's not sensory driven, but it's driven by patterns of activity. What kinds of patterns of activity? We have the visual system in mind. Well, patterns of activity that are exactly these retinal waves. So what we did is we actually took a model of retinal waves published in 2007. So here's a model of what these retinal waves look like, just very similar to the patterns that we saw recorded experimentally, so propagating throughout the entire retina. So this is now actually the entire retina, so that's why they look more patchy, even though they're really the same retinal waves as the ones we saw experimentally. So we have the retinal waves, and the first question we had was, okay, how do we go from retinal waves to actual activity patterns on the dendrite? So luckily for us, there was a study a few years ago from the lab of Sonja Hofer that measured receptive fields of these individual synaptic inputs. This was actually done in the adult cortex. So here are these individual synaptic inputs on a dendrite in these red circles. And using sort of noise and reverse correlation analysis, they were able to describe the receptive fields or the filters that describe how these synaptic inputs process inputs. And they found that they're Gabor filters. I'm pretty sure you've touched upon Gabors and the idea of Gabor filters previously in the course, but let me still describe to you what they look like. So Well, first of all, they are localized in space. So what that means is that each individual input only looks at, so if you look in the visual world, they only look at very small portions in the visual world, right? So each synaptic input doesn't look at the entire, doesn't code for the entire visual scene, just codes for a portion of the visual scene. So in these rectangles here, where we describe each of the receptive fields of the synaptic inputs, 20 degrees is basically the angle, visual angle. So you see that these patches, these Gabor patches are localized, right? They're not spread out through the entire rectangle, but they're localized. Importantly, they consist of a positive and a negative component. So that means that upon the presentation of some stimulus, parts of these part will be excited, part will be inhibited. That's what the red and the blue denote, and they are oriented. This is super important. So you can draw basically these lines. You can draw a line here. This is about minus 45 degrees. This is a vertical, so 90 degrees, and so on. So this is the model then that we had of these receptive fields, these Gabors. So we modeled the center in visual space. So where exactly in the visual field this particular input
Can I
is
ask a
looking
question
at.
here?
Let me just finish, yeah. And then we also modeled the orientation. So basically they're all oriented. As I said, you can kind of draw a line through the positive and the negative field. And so we had the angle to denote the orientation. Yes, ask me.
So here in the right bottom here, right, 20 degree angle, those are experiment data, right? And those are how excited neuron and inhibitor neuron group arranged in the cortex, like just exactly like with the angle.
Yeah,
like
so
they're in between.
No, so these are the synaptic inputs onto. These are all excitatory. These are all excitatory synaptic inputs. So each of these 1, 2, 3, 4 are all excitatory. The reason that we know they're excitatory is a little subtle. You see that they're all protrusions outside of the dendrite. So they're called dendritic spines. And so we know that excitatory inputs usually form through these spine protrusions on the dendrite. So they're all excitatory inputs. Inhibitory inputs actually primarily form without these protrusions. So they form just on the dendritic branch, and they're known as so-called shaft synapses. And they're a bit more difficult to quantify. But these protrusions, because they're protrusions, they're very easy to kind of spot, and so you can find them. So all of these are excitatory. The reason that I say they have kind of a positive and a negative component, so the shape that they have in this Gabor really is, again, a old Hubel and Wiesel idea. It just basically says you have inputs that come from the LGN to the cortex, and so in like the very simplest scenario, a Gabor receptive field in the cortex would be formed by lining up several receptive fields in the thalamus, and in the thalamus the receptive fields are center surround. So there's basically a Gaussian that's positive or negative in the middle, depending on whether it's on or off, and then the surround is usually of the opposite sign. And so if you align several of them properly, you can basically generate this Gabor patch in the cortex. So the reason that you have this positive and negative part of the Gaussian is because of how the positive and negative part in the center surround receptive field in the thalamus form.
I think it's clear.
I hope that's clear.
Yeah,
of course, we don't know that these synaptic inputs are all coming from the thalamus. But it's not important. So we don't know. I mean, in the model that I'm showing you now, we are saying, okay, we have the retina drives the thalamus drives the cortex. So it's all kind of like a feed-forward setting. But this doesn't have to be the case, because we're modeling these synaptic inputs, or the synaptic inputs that they characterized in this Yacaruso study. And we don't know whether the inputs that are received here are coming from the thalamus or coming from other cortical neurons. But yeah. So the kind of the explanation that I gave you was purely about the feed-forward projections. But I mean, the same principle also holds for recurrent connections, because if we know that a single cortical neuron is orientation selective because it can be described by these Gabors, then if it provides input to another neuron, then it will also be orientation selective. So So that can be also reconciled.
So how many of these synapses, or what the projective field, I would say, maybe are visual stimulus orientated? Maybe there are other synapses as well.
Yeah.
Features.
Yeah. How many are actually oriented? I don't really know. I think most of them are. Yeah. I mean, okay. So this comes in the issue of. So we know, for example, that simple cells in the cortex are orientation selective because you can we understand how they generate their receptive field. Complex cells are more complicated. So I don't know off the top of my head what's the ratio of complex versus simple cells. And also I don't know if you're looking at a simple cell, what the fraction of inputs that it receives that are orientation selective versus not. I don't know.
I see.
I don't know that people have really.
There's a broad spectrum. There's a broad spectrum in monkey, and there's a broad spectrum in mouse. But it's slightly different because in monkeys you have hypercolumns, right? So then the recurrent connections are mostly oriented in the same fashion, right? But in the rodents, there's no map. Right. So then I think the current connections, these spines should be all sort of random. So, or at least broadly distributed.
Yeah, but I'm not sure that you would say that they're random. You don't have the map, but we know also from the work of Mercedes Flogel and Sonja Hofer, right, that even though you have the salt and pepper distribution, so now we're kind of
it's true.
But you still have strong connections between neurons that are similarly oriented. They may be spatially far away from each other, right? If you, they don't necessarily form the map, but they would still be strongly connected, right? So, yeah. So I would say, I mean, these are all interesting questions, but yeah, I'm not an experimentalist. I don't study the visual cortex and I don't do these experiments. So I think what I want to kind of convey to you is that we can describe the organization of these synapses using theoretical models of activity-dependent plasticity. So I will just focus on that. I mean, of course, these are very interesting questions, but yeah, maybe let's leave them for the end when other experts can chip in. Okay, so let's just remember where we're heading. So we want to understand how these synaptic inputs get organized. So first I showed them to you with black circles. And the first kind of component of the model is how to drive these synaptic inputs. And what I'm trying to tell you now is how can we go from these natural inputs that are the retinal waves to spike trains or burst trains on each of these individual synaptic inputs. And the way we do this is we start with the retinal waves. We implement what's called the linear nonlinear model. Maybe you have studied this. I didn't want to mention it, but first we linearly filter the receptive field, the retinal waves with receptive fields that are these gabors, because these gabors have been found experimentally. And then once we linearly filter them, we pass them through an exponential nonlinearity and then use a Poisson spike generator to basically generate activity patterns on these individual synaptic inputs. So we effectively have a transformation that go from retinal waves into spike patterns on the individual synaptic inputs. And from now on, I'm going to color these synapses. So they're no longer going to be black. The color here is going to denote this orientation, this angle of orientation that I show you here. And I'm going to use this color wheel to tell you what that orientation is. So for example, yellow means a horizontally oriented receptive field. So the first ingredient of the model is exactly what I just described. How do we go from retinal waves in the retina? So this is the sensory periphery. They clearly propagate to the cortex. So how do we go from these retinal waves into activation patterns of these synaptic inputs in the cortex? And we do this via these Gabor receptive fields because of experimental evidence. The second and more relevant for today's lecture aspect is, now that we know how to drive these synaptic inputs, we need to understand how to use these activity patterns to reorganize the connectivity in the in these dendritic branches. So we're going to propose an activity-dependent plasticity model, very similar to the ones we talked about on Monday's lecture, to now understand how these activity patterns are used to organize the synaptic inputs. So I'm going to describe to you the phenomenological model, and then once you understand hopefully what this means, what this does, I'm going to cover for you a biophysical implementation. If you care, you can also space out for that bit. But I just want to tell you that we have a phenomenological understanding for what goes on, and we also have a biophysical implementation for how we think that this mechanism can be implemented. I think this was a bit of the criticism in Monday's lecture when I talked about all these Hebbian mechanisms, was exactly how could these mechanisms be implemented. So we think that in this context, we have a good idea for all the molecules that could implement this mechanism. But let me first tell you what the mechanism is. So we have a mechanism that is very much the flavor of Hebb. So we look at the small portion of this dendrite. So let's take this one where 3 synaptic inputs impinge on this part of the dendrite. And we assume that retinal waves pass, activate these synaptic inputs as we described before. And we now need to understand how these synapses can become potentiated or depressed. So let's assume there's a retinal wave that passes in a particular orientation. They will preferentially activate synapses that have close to this vertical orientation. So these would be these 2 purple synapses. So notice now, rather than the red and the blue, I'm going to use the full and the dashed line ellipse to denote these positive and the negative parts of the Gabor. So you see, if we draw a line, in both cases, it's going to be vertical orientation. So these 2 synaptic inputs will become activated, and via the Hebbian mechanism, they will become, they're coactive, so they will become potentiated. So we call this correlation-dependent cooperation. So these 2 synaptic inputs experience correlated activity because the retinal wave activates them at the same time. And as a result, they cooperate and both become potentiated. The second ingredient in the model, in this activity-dependent refinement, is distance-dependent competition. So this tells us in which context other synaptic inputs will become depressed or basically outcompeted. And it becomes the synapses that become outcompeted are the ones that are basically orthogonally oriented to the ones that are activated. So if we have a retinal wave that activates the vertically oriented synapses, then the horizontally oriented ones will not be activated. According to Hebb, if they're not activated, they should become depressed. And we do this in a distance-dependent fashion. So the closer the synapse is, the more influenced they will be by the cooperation of the activated synapses, and is more likely to be depressed. And the farther away it is, the less influence it will have from the activated synapses, and it's less likely to be depressed. If, however, is depressed and eventually eliminated, we have the third ingredient in the model, and that is structural plasticity. So that means that whenever a synapse gets eliminated, we basically put a new synapse on the dendritic tree in a random location and with a random orientation. And then the process repeats. The motivation for the structural plasticity is again based on experimental evidence, because it's sort of like a normalization or homeostatic mechanism that enables the preservation of the same number of synaptic inputs. So the density on the dendritic tree is preserved. If we didn't apply this constraint, we run into the problem that synapses get eliminated, and then we basically prune out synapses and not many synapses remain on the dendritic tree. And then the process repeats itself. So we now have basically these 3 ingredients. So synapses can get potentiated if they're coactive. Synapses can get depressed if they're, and outcompeted if they're not coactive with their neighbors. And this is done in a distance-dependent fashion. And then every time a synapse is eliminated, a new one is born. Are there questions? No questions. Okay. So I said we have an understanding for how this could be implemented basically biologically or through interaction of different molecules. So the molecules that seem to be involved in this process and development are known as neurotrophins. And this was basically discovered by, again, my collaborator, Christian Lohmann, who I mentioned discovered the clustering in the first place in development. So then they had a follow-up paper a few years ago where they exactly pinned the process down to these molecules called neurotrophins. And there's two flavors usually of the neurotrophins. Ones that are kind of the positive neurotrophins that regulate the emergence of new synapses, their potentiation, their stabilization. And there's negative synapses called negative neurotrophins called proneurotrophins that are usually responsible for the synapse elimination, for their depression, for their decay. And in this context, the two neurotrophins that are involved are BDNF, so brain-derived neurotrophic factor, and pro-BDNF that end up binding to different receptors and are responsible for the generation of these positive and negative actions. So BDNF is the positive one, binds to track B and then causes potentiation, and pro-BDNF is the negative one, it binds to P75 neurotrophic receptor and causes depression. Now, the really cool thing is that in the Lohmann group, they identified effectively a transition molecule, this MMP9, matrix metalloproteinase 9, that ends up converting one to the other molecule. So you basically have a toggle, a switch going between depression and potentiation, because it seems like early in development, there's more of this depressing molecule, this pro-BDNF, but if you have a lot of activity in cooperation, so coactivity between synapses, you can convert the pro-BDNF into BDNF. And so you really have a mechanism to kind of balance out the potentiation and depression. Okay, so that is really the mechanism that we have that can generate these phenomenological descriptions that I showed you on the slide before. And now the interesting part comes to understand, we have a way to go from retinal waves into activity patterns. We have an activity-dependent learning rule that has a very concrete biophysical implementation for how these synapses should be refined. So now we want to put this to test, simulate a developmental scenario of potentially this activity-dependent organization of synaptic inputs. So we're gonna see what happens. We start with a scenario as during birth, so postnatal day 0, where we distribute these randomly located and randomly oriented synaptic inputs on the dendritic tree. We simulate the retinal waves and apply this plasticity rule and wait for 2 weeks, just like in normal development, and we wait to see how the synapses organize themselves. And what we find is that the synapses organize themselves in a way that they become clustered according to orientation. So what you see is we now have this cluster that corresponds to the pink synapses. We have a cluster that corresponds to the blue synapses, the green synapses, the dark purple synapses, and so on. But you don't just have to believe me. By looking at this example, we can actually quantify this. So we can measure the same variables that they identified in the data. So we can measure the coactivity or the correlation of neighboring synaptic inputs as a function of their distance. And what we find in this model of spontaneous activity is that this coactivity is high for inputs that are near each other and then decays as a function of the distance between the inputs. If we now don't drive these synaptic inputs with this structured spontaneous activity in the form of the retinal waves, but we actually drive the system with noise, so just pure white noise, we can completely eliminate this relationship and we basically get a flat curve, the curve that we see at the bottom. So we don't get clustering. And I just want to compare this data to orientation clustering in the ferrets. So the data points here are shown in the green. These were experiments done in the lab of David Fitzpatrick, who found exactly this type of orientation clustering that we also identify in the data, but in the adult ferret. So what we'd like to propose is we have a mechanism by which this clustering can emerge in development. Of course, in our model, we don't know eventually whether these synaptic inputs will be the same ones that are selective to orientation later in adulthood, but that's what we think we have come up with. The next interesting thing that I want to point out is to do a comparison across species, because we already had a mention from Louis earlier that there is indeed difference in the kind of global organization of inputs in the ferret, not in the ferret, I think that he mentioned mouse, he mentioned macaque, so monkey, as well as rodents. So ferrets are kind of like monkeys in that sense. They also have this map of orientation selective neurons, unlike the rodents, which are salt and pepper. But because here we're talking about subcellular organization, there seems to be a difference in the subcellular organization between the ferret and the mouse. So in the ferret, we indeed have this orientation clustering that is basically the result of our model. So it's predicted by our model. And the reason that we get it is that if we look at these neighboring inputs, synaptic inputs that become coactive or correlated, so for distances that are small, we see that these neighboring synaptic inputs effectively get lined up so that they really share the same orientation. So that's why we get this so-called orientation clustering. But in the mouse, this does not seem to be the case. In fact, in the mouse, just like these maps, we also see a difference relative to how the subcellular organization is realized. In other words, we do not have orientation clustering. And yet, in the mouse, we still get, I'll show you in a moment. Okay, I'll show you to you here. In the mouse, we still get the correlation versus distance curve that is indicative of synaptic clustering. So what is going on? So how can we have clustering in the mouse, so functional clustering in the mouse, but not get orientation clustering as we do in the ferret? And the answer for this actually lies in the sizes of the cortices. We think it's actually related or similar to the explanation between why you get maps versus salt and pepper patterns. And the reason is in the sizes of the cortices. So in both animals, it looks like the neurons in the cortex have about the same size. So the length of these dendritic branches is about the same. But the ferret cortex is about 30 times bigger relative to the mouse cortex. So here we see the one scale in one direction, but if we look at the area, it's about 30 times bigger in the ferret. So this is like relative comparing the mouse cortex to the ferret cortex. And because of this difference, so the neurons are the same, but the ferret cortex is much, much bigger. If we take the perspective of a single excited tripyramidal neuron in the cortices of these two animals, we basically have a scenario where in the ferret, we have a very, we have magnification of visual space. So let me put these blue contours in the visual cortex as an indication of degrees of visual angle in the visual world. So basically in the ferret, if we take this reference pyramidal neuron as the neuron we're looking at, and then we look at what other inputs it receives inputs from, what other neurons it receives input from, we see that they're distributed in a very small region of visual space that's basically indicated by these blue contours. So the blue contours just tell us how many degrees of visual angle you look at in the visual world. So this reference pyramidal neuron really receives inputs from a very small region in visual space. So you have a huge amplification of visual space. In the mouse, we see that this reference pyramidal neuron will receive inputs from neurons that are very far away in visual space, right? This other neuron is, I don't know, 10 blue contour lines away from the neuron that's nearby here. So I forget, maybe I think this is about 7 degrees of visual space. And so here, you know, one pyramidal neuron will receive about 7 degrees inputs that are at most 7 degrees different. But in the mouse, this will be like, you know, 50 degrees or 70 degrees of visual space. I don't know, it's not probably 10, but let's say 50 degrees of visual space. So that's why in the ferret, we have this huge amplification of visual space. And in the mouse, you know, one neuron can really receive inputs from basically other neurons that look at all other regions in visual space. So because we have a difference in this amplification factor in the mouse and in the ferret, we have a very different result into the clustering that emerges in these two different animals. So in the ferret, neurons that are nearby will become orientation selective because this is how their nearby receptive fields line up. But in the mouse, you generate these high functional correlations for nearby inputs, not because the neurons become orientations clustered according to orientation, but they actually end up being clustered according to overlap. So here's an example of two receptive fields. This is just a schematic. of two receptive fields that are nearby each other, you see that they're completely orthogonally oriented, and yet they still exhibit high correlated activity. Why is that? Because they overlap. So you see that the red regions now of the Gabor receptive fields line on top of each other. So when a retinal wave passes, in the case of the orientation clustered neurons, they will all get coactive because they will be, if the retinal wave is appropriately oriented, then they will just get coactive because they all have the same orientation. In the mouse, even though they shared very different orientation, they will get coactivated simply because the receptive fields overlap. So you can still generate these high amounts of coactivity despite the fact that you don't have this orientation clustering. So we think that we have come up with a developmental model that can explain elements of clustering with regards to very different sensory features in the adult animal. In the ferret, the sensory feature in adulthood, according to which the synapses clusters orientation, and in the mouse, the sensory feature with which they cluster is now overlap. Are there any questions? This is like the biggest conclusion that we have from this work. So we have, it's this exactly same developmental model. Everything is the same. So the same retinal waves, the same activity-dependent plasticity mechanism driven by these interactions between BDNF and pro-BDNF. The only difference that I have applied here is difference in the amplification factor between the ferret and the mouse. So basically what I mean here is what I end up doing to capture this difference in amplification factor. Let me go back to this one. Remember, I model each receptive field with an orientation and with a center. So to capture the fact that in the ferret, they're less dispersed than in the mouse, they're more dispersed, I literally just take these centers. So here's visual space, right? This is degrees in visual space, this entire box. So in the mouse, I will just scatter them everywhere because they're more dispersed. And in the ferret, I will just focus them in small regions. So that is the only difference that I've applied, is how I have scattered, or how Jan did this work, how Jan scatters the centers of these receptive fields. So to capture this difference in amplification, in the mouse, you just scatter them everywhere in visual space. In the ferret, you put them more localized. Ask me, Zushi.
But the question is whether this also agree with the natural images, right? The way that you orientate those edges, right? You cluster them and you're spreading them out. The statistical match what we the mouse.
Yeah, absolutely. So we definitely we took the data about how spread out these receptive fields are from data. So we exactly used so this idea that the ferret does the amplification. We took the spread, how spread out they are in visual space for the ferret versus the mouse. We took this from experimental measurements. I think you can look up the details in our paper, but we actually have kind of like we fit the spread function basically of each of the two animals, and that's what we use in the model. And the issue about natural images is a bit different. So I can address this maybe later, but I just want to point out that here we're not dealing with natural images because we have a developmental model, and we know that it's like these retinal waves that are driving these refinements and the emergence of this clustering. But there is, one could ask the question of, okay, you observe these two different types of clustering in the ferret and in the mouse. So one could then ask, what are the functional implications or benefits for this clustering in terms of how then images are processed? So I can comment on that, because this is all developmental. So we think this is all, it could be, we think that this could be set up very early in development before animals can even see natural images. And then the question arises, okay, if it is set up developmentally and we have this already set up at eye opening, then at eye opening, the animal all of a sudden starts receiving visual input, sees natural images. Can this somehow be helpful in processing certain things in the natural images even though the animal, hadn't seen natural images before.
Mhm.
So can this clustering be helpful in how natural images are processed? And the answer is yes. But there, yeah, I can comment on this in a few slides if you want. Zoshi, what was your question? Was that your question?
Yeah, my question here, I think you're probably going to mention in the later when you describe the model, I guess, because I wonder how we define receptive field here in the mice case. Then I mean they're all overlapped and co-activate. Do we just say, okay, this chunk is a receptive field, and how do we further refine them? Say under this chunk, the subgroups. Yeah, like how do we define them?
I mean, we define them, the receptive fields are defined as I defined them before. So
orientation.
Yeah, so as in here. So each of these circles basically can be described by two Gaussians, a positive and a negative Gaussian. And there is a center. And the center is basically a coordinate in visual space, so azimuth and longitudinal or orthogonal. So it's basically a coordinate in XY in visual space. Because, right, so we're talking about a dendritic, we are talking about a neuron. The neuron has dendrites, and the dendrites has synapses. So we look at the single synapse, and this particular synapse codes, right, responds to only a single location in visual space, right? Everything is localized, right? So this neuron is not going to respond to everything, or the synapse is not going to respond to everything that you see when you look out, right? So
it's a pixel.
It's only going to look at a single point in visual space. And that point in visual space is described by these coordinates XY, which denote like the center of the receptive field of that synapse. And the synapse also has a preferred orientation. So that when a retinal wave passes, right, the retinal wave, so we can put a square around this circle. Sorry, this is circle, but it can also be a square. And each of these pixels in this circle/square is also matched to a particular XY location in the visual world. So we think of these retinal waves effectively as mimicking, natural images that will eventually be presented to the animal once the eyes open. So each of these pixels here in the retina that are experiencing the retinal waves indeed corresponds to an XY location in the visual world. So then if I take a synapse, let's say I take this green synapse, and the XY location in this green synapse is, I don't know, 15 and 20 degrees, 15x and 20y, that could correspond, for example, to this, let's say to this location where my little hand points. So that every time a retinal wave passes through this location, you will activate that synapse. But you will not activate the synapse that has an XY location, for example, down there, because obviously you don't have activity down there when a retinal wave passes up here. So that's the model. Yeah, and then the difference between the mouse and the cortex is simply in my initial setting, and then every time I go through this process of refinement, right? So I do this refinement. So I eliminate the synapse and I bring a new one, but also at the beginning, when I distribute these synaptic inputs on the dendritic tree, and whenever I bring a new one after a previous one has been depressed, basically how far I scatter the XY. So let's say I take this green synapse looks at this particular position in visual space. Then the ones that are around it in the ferret will also, so this red one, this blue one, this purple one, they will have XY locations that are around in this area. But in the mouse, they will cover a bigger area. So they will be more spread out. That's how I model it. So that's the only difference in the model. Otherwise, they will experience the same retinal waves. They will experience the same type of refinement according to this rule. Everything will be the same.
Yeah, this is
okay.
pretty clear. Thank you.
Okay, super. Okay, so the first point that I wanted to convey to you is that we have this difference in kind of the local patterns of clustering. So we have the orientation clustering in the ferret and the overlap clustering in the mouse. So I'm going to show you a few more slides and then I'm going to take a break. I want to just show you that we don't just do this for the ferret and the mouse. We can in fact extend this to other animals because we don't necessarily know how spread out the receptive fields are. This hasn't been measured beyond the ferret and the mouse, but we do know how large the cortices are. So we can infer the spread of receptive fields in other animals. So Jan did this for rabbit, cat, and macaque. And so one can now basically measure, or I can put in the axes the receptive field spread. There seems to be a slight difference in diameter. I didn't mention this parameter before. It's very small, so it's a not we don't think it's like super important, but anyways, like in the mouse and the ferret, there's not such a big difference in the diameter, but this came up in our reviews, so we did this analysis. We think the more important parameter is this like spread of receptive field. So what I was saying, where the XY location is in the mouse versus the ferret versus the other animals. And so based on this, we can sort of predict what type of organization you will have in other animals. So where the rabbit and cat and macaque will go. And there might actually be people in the audience who studied this, but there was a group that published recently, tell me if you're in the audience, who was a study last year, I think, or maybe even earlier this year in Nature Communications that actually found in macaque, we were right, so this came out as our paper was in review, that in macaque, indeed, green corresponds to orientation clustering. There is orientation clustering in macaque. There's also clustering with regards to other features, color and other sensory features. So, you know, part of our predictions, which ended up being postdictions, are right. Okay, so we can go beyond even the existing data and we can make predictions for other animals. Maybe we take a break now. I was going to take a break actually as I move into the second topic, but I'm not sure if you guys have patience to hear me.
Yeah, we can take a break. Yeah, it's totally
take a break.
fine.
Okay, so we come back in 5 minutes, maybe stop recording and we come back.
Sure.
Super. Okay, I am back. If everybody else is back, or we take, I think it's been exactly 5 minutes.
Yeah, maybe I can ask you a few more questions before you
I
continue.
think you're muted, and I don't hear you.
Maybe I can ask you a few more questions, maybe before you continue.
Sorry.
Can you hear me now?
Yes, I can.
Okay. One is just a summary that, so you're saying that for the mouse, what happened is that you have this clustering of spines, but they can be physically, the receptive field can be physically spreading out, right? They're and they can have different orientations. Yeah. But for the monkey or the other animals, the clustering is mainly due to the same orientations, right?
Yeah.
For nearby spines. Okay, that's
Exactly.
if I understand correctly. But what okay, but you also have a procedure that you prune some of the synapses, right? Those that are not activated, are not co-activated, then you take them away, right? This is, yeah. But so now in the end, you have this whole tree, the whole dendrites that have different orientation. Then what determines the orientation selectivity of this postsynaptic neuron? Yes.
Yeah. Good question. Yeah, so in the second part of this, it will be very quick. But what I want to next move on to is exactly this question that Trent is now asking. And that is, so everything I showed you is local, small portions of the dendritic tree, about 100 to 150 microns. That's basically what we've been modeling here. But the question now is, what happens on the entire dendritic tree? And what I first want to answer for you now is, what does this say about the global organization of these synaptic inputs on the dendritic tree for mouse and for ferret? And the question that Trent's asking, okay, what about how does this set up or determine the selectivity of the soma? I'm not going to really answer the question about, excuse me, about the soma. And the reason is it's a little bit complicated. So I will show you a simple model for how the soma integrates the inputs from the rest of the dendritic tree. And I'll comment a little bit about other ideas and models for what the soma should be doing. But now I want to focus on what this local organization, so we obviously have a big dendritic tree. So far, we've been looking at these small dendritic portions. So what does this local organization imply for the global organization of synaptic inputs on the entire dendritic tree? All right, so yeah, we're going to look at the entire dendritic tree now. So here is an example of how, in our case, the soma will be integrating inputs from the entire dendritic tree. So we assume that all the inputs are effectively combined at the soma in a linear fashion. So inputs that are far away have less influence than inputs that are nearby. Well, actually, I think we're just summing them up with equal influence, because I think it's known that inputs that are far away have stronger amplitude, so that their effect at the soma is similar to inputs nearby. So I think in this case, we have a super simple model where we're just adding up the activity of all of the inputs to determine the activity at the soma. And this is the signal that it generates at the soma. So we have these very high periods of high activity, these short periods of high activity. We then threshold that, and whenever the threshold is crossed, we basically say that the soma generates an action potential. Now, this is actually a more complicated process than the way we model it. This is work in progress. There are indeed certain amplifications, nonlinearities, whatever, NMDA classifiers that are generated in different parts of the dendritic tree. There's some recent studies from the Fitzpatrick group led by Ben Scholl, who's a study earlier this year in Nature, that actually says how different small versus weak synapses influence the ultimate orientation selectivity of the soma. But I'm not going to go into that. You can ask me for the references later. But the important point that for now is that everything is just linearly summed up. This gives us a signal at the soma, and that signal crosses threshold, we assume that an action potential is generated at the soma. So when an action potential is generated, we have a back propagating action potential, as found experimentally many years ago, that tends to go back to all the dendrites in the dendritic tree. So we know that this back propagating action potential decays, so it's more strongly sensed at synapses near the soma than at synapses far away. So here's an example of this decay as a function of distance. And so if we now include this model of spike generation at the soma and back propagation of this action potential to faraway synapses that decays with distance, what we now see is that synaptic inputs on the entire dendritic tree end up organizing themselves in a global manner, so that synapses that are nearby the soma acquire similar selectivity to the soma, and the ones that become far and far away end up just being, they're still clustered locally, but their orientation selectivity ends up being completely unrelated to the orientation selectivity of the soma. So it's a little bit difficult to see from this one example. It's a little bit easier to see if we average over multiple dendritic trees. So here's an average that is shown here at the very edge of the slide. The parameter that we have varied here, indicated with the different shades of orange, is the strength of this back propagation potential. So for now, let's focus on the middle plot. So what we have here is a scenario where we plot the difference of orientation between the soma and any other input. So the difference is low, meaning that the inputs share the same orientation as the soma if the color is bright. And the difference is high, meaning that the input has effectively a random orientation relative to the soma if the color is dark. So you see now that we have this gradient of color from bright to dark as we go far away from the soma into these peripheral branches on the dendritic. So what this means is that, as I already summarized for you, synapses that are near the soma have a small difference in orientation relative to the soma, meaning that they share the same orientation as the soma. But if we look at synapses that are far away, they share a very big difference to the soma, so 45 degrees, effectively meaning that they're randomly oriented. They have basically no correlation. Their orientation is not correlated to the orientation of the soma. Now, this gradient of global organization of synaptic inputs depends on how much these back propagating action potentials decay. So if they decay more slowly, we see that more of this difference is bright, meaning that more inputs near the soma share the same orientation as the soma, and only the ones that are really, really far away will be different than the soma, and vice versa. If we have a backpropagating action potential that decays very fast, meaning that its effects are not felt as you go far away from the soma, we basically have a dendritic tree where we don't have any order. So we have still the local order, but globally, almost every input has nothing to do with the soma, except maybe for the inputs that are very, very close to the soma. So a summary from this part is, this is the ferret model where we know that we have this local orientation clustering. If we now look at how globally the synaptic inputs cluster and we add this element, namely this backpropagating action potential that can induce this global order, we see that indeed we also have global organization of synaptic inputs, and they're globally organized in a way that the synaptic inputs that are near the soma end up sharing the orientation preference of the soma, and those that are far away end up having basically independent orientation preference relative to the soma. Is that clear? So that's a prediction. We don't know if this happens in the data, but there is a small element in the data that we can kind of anchor this prediction to. So let's look at some data from the Fitzpatrick lab.
Is there any learning rule involved when you talk about the backpropagation?
No learning rule. It's the same. It's exactly the same learning rule. We still have the same learning rule that sets up the local order, but now in addition, we introduce the backpropagating action potential. So it's the local rule in combination with the global backpropagating action potential that sets up this global order. So kind of the local effects of the learning rule get globalized because of the BAP. Of course, only if the BAP has the slow decay. If the BAP is local, then again, the locality remains. The locality of the learning rule remains. So what we can do is, sorry, I'm gonna just flick the next thing. So in the Fitzpatrick lab, unfortunately, they did not, it was just difficult for them to basically look at the distribution of synaptic inputs on the entire dendritic tree. But what they did is, curiously, out of all the branches that they analyzed, so they could only analyze branches and clustering on single branches, but they were able to divide up their branches into two types. They call them clustered versus heterogeneous branches. So the clustered branches were branches where they basically saw like one big cluster of synapses. So all the synapses have shared the same orientation, or maybe they had, if the branch was, maybe they had two big clusters. And then they saw heterogeneous branches where basically all the synaptic inputs were just not there. They were just all over the place. They were kind of random. And so to kind of visualize this division, they plotted the cumulative density function as a function of circular dispersion, which is basically the difference in orientation between the synaptic input at the soma. And so, this is how, in fact, they define these clustered heterogeneous branches. So in the clustered ones, you see that, yeah, you really clusters. So that means that you have a lot of inputs that share the same orientation. That's why the circular dispersion is low. Remember, the dispersion is just the difference in the orientation. And in the heterogeneous ones, you have much, so the CDF, the cumulative density function, is much flatter, meaning you just have almost random orientation of nearby synaptic inputs. Now, in our model, we have this global prediction of order. We could capture these same clustered versus heterogeneous branches, just splitting up our branches into two categories, purely by whether they're proximal or distal to the soma. So we said proximal branches are ones that are less than 150 microns, and that are bigger than 150 microns. And if we overlay them on the branches, we could basically find a very nice match, suggesting that our model predicts this global organization of branches, and to match them to the data, we propose that the clustered branches that the Fitzpatrick lab model identified in experiments would primarily be proximal, and the heterogeneous branch that they found would primarily be distal. So this is a prediction. We don't know if it holds. This is what data seems to suggest. So that's what happens in the ferret. We have local orientation clustering, and if we introduce this global backpropagating action potential, potential, we can also generate global organization of synaptic inputs, where near inputs to the soma also end up sharing the orientation preference of the soma, and far inputs do not. What about the mouse? So in the mouse, we do not have this local orientation clustering, but we have what we called overlap clustering. So what this ends up generating is a different type of global organization in the mouse. And we actually call this a topographic map that is imprinted on the entire dendritic tree of the neuron. So we still have some kind of organization, but here the color no longer denotes similarity of orientation of the synapse with regard to the soma, but here the color denotes basically the difference in this X location in the visual world between the soma and the synapse. So again, it's a function of this decay of the vector gain action potential, because this is the globalizing signal that acts in addition to the local synaptic plasticity. So if we take the plot in the middle, we see that if we now have some intermediate level of decay of back-propagating action potential as you get far away from the soma, we get synaptic inputs near the soma, end up having effectively XY location of their receptive field very close to that of the soma. And synaptic inputs that are located in the dendrites end up getting XY, they have an XY location that's very far away, XY location of the soma. So they really look at very different portions in the visual world. On the other hand, the inputs that are near the soma look at the same portions of the visual world. And if we change the properties of the back propagating action potential, we can either completely homogenize the entire dendritic tree, or if back propagating action potential is local, we can just get this effect near the soma. And we understand why this happens. I don't want to go into the details. I will just mention, if it doesn't make sense, you don't worry too much about it. So basically what we get is, this plot here illustrates an initial, this plot here demonstrates a final scenario. So remember I said we start with a bunch of Gabors where the centers, so a bunch of synaptic inputs where the centers of each synaptic input are quite spread out. So this is the mouse. So these are the X of 3 of the Gabors. So this Gabor has this X, this Gabor has X, and this Gabor synaptic input has this X. So they're quite spread out. This is the mouse. And then via the process of synaptic plasticity, the final configuration ends up being like this. So they end up organizing themselves into this structure. We call it like a petal structure. You see that they all line up. Remember, the local mechanism makes them all overlap. So they all end up overlap. So all of their positive parts indicated here by the full line ellipses end up being focused in the middle. So there's really like a large overlap. And the negative parts of the ellipses go around like a flower. And because of this, we sort of have a little bit of a contraction of these X's. So the X's, the centers of the receptive fields, are no longer as spread out. They end up being slightly contracted to a small area. And this is what effectively generates this difference in the, this is what effectively generates this global map, a global topographic map of the receptive field centers along the entire tree. And we can measure this. So we can exactly measure this for the 3 different decay paths of the back propagating action potential. And so relative to the soma, how far away the XY location of the synapse is relative to the soma. So 25, or this is the chance level. And we can basically compare that to the data. So this is data from the same Iacaruso paper that found these Gabor receptive fields at the synapses. And it looks like this intermediate decay profile of the back propagating action potential, this middle orange curve, corresponds quite well to the data. So this idea that we get visual topography was actually found in the data before we proposed their model. But we weren't after capturing this result. We started with, with our goal was to capture just this local clustering that I showed you at the very, very beginning. And then we saw that our model can explain a lot of different things, even though initially that was not our goal. We were I don't want to say fortunate, but we saw that the model is actually quite powerful in explaining a lot of these different effects, even though they were not kind of the initial goal of the model in the first place. So yeah, so we do think that even though a lot of these phenomena are observed in the adult animal, they're probably set up developmentally by this very simple activity-dependent plasticity rule that has this biological motivation of the neurotrophic interactions and these the non-random activity patterns from the retinal waves. So I have basically next my summary slide, and I want to move on to the next topic. But now I want to ask if there are questions. The summary slide just tells us that we have proposed a learning model that can generate both local organization in small parts of dendritic trees and global organization. The local organization that I showed you was driven by the retinal waves and by this local plasticity rule that potentiates nearby synapses and depresses faraway synapses that are uncorrelated with the nearby synapses and can generate either orientation clustering in the ferret versus overlap clustering in the mouse purely because of this amplification factor. So the sizes of the cortices and therefore the spread of the receptive fields. And then everything remains the same. If we just add this global backpropagating action potential, we can also generate global organization, which in the ferret is this orientation gradient of synapses. So nearby the soma, they acquire the same orientation as the soma, far away, independent of the soma, or this retinotopic or topographic gradient, if you want to call it, in the mouse, where synapses that are nearby end up looking at exactly the same point in visual space as the soma, and synapses that are far away looking at a different location than the soma. So yeah, I'm happy to take questions. And if you want, only if you want, I can also mention something about the visual implications of this orientation, of this types of clustering.
So in the ferret, there still is some renotopic gradient, right?
Yeah. In the mouse, only in the mouse, not in the ferret.
Only in the mouse, so the ferret doesn't.
Yeah.
Ah, okay.
Exactly. So this is data from the mouse.
Can you give us some sort of intuition that how this global signal and the local signal interact, which in the end you cause this new arrangement of the receptive field? So let's take, for example, this overlap clustering, right? You nearby spines, they can have different orientation, but they can be physically far away, right? This is individual world. But then in the end, after this global signaling, it becomes that the receptive fields are also physically clustered together. Did I say it correctly, or I missed something?
So in the mouse, you don't have orientation clustering, right? So locally, the synaptic inputs are not clustered according to orientation. Yeah. They just end up being clustered according to overlap. Yeah, maybe this is the schematic to show. So this is like the initial scenario. We basically put the synaptic inputs, so they're spread out. So the X's are the locations in visual space of each of the receptive fields. So when I say Gabor or I say receptive field, I really mean the same thing, or a synaptic input. So the X's are the centers of the Gabors for each receptive field for each synapse. So this is the initial scenario where they're spread out. Mm-hmm. And then we get a combination. We have a local learning rule that is giving us this local overlap. So these are, let's say, local. And so what the learning rule locally does is it makes them all overlap. That's the local effect. So we get the scenario where they end up overlapping, right? So you see that all of the positive parts indicated here by the full ellipsis end up lying on top of each other to overlap and get this flower-like petal structure. And notice now I haven't put the X's here, but if I put the X's here, you will see now that the X's are in a smaller region of spatial space than they are here. Here, they're more spread out than here. So we call this like some kind of a contraction phenomenon. So the local learning rule that generates this local overlap clustering meaning that if we just look at small portions of dendritic tree, the synapses, they're not going to share the same orientation, but they will still show large amount of functional correlation because their receptive fields will overlap. So the generation of this local overlap will cause this contraction. So this spread of the centers will become smaller. And because the spread of the centers becomes smaller, and we now introduce this global backpropagating action potential, we end up effectively kind of contracting the receptive fields of synapses that are nearby the soma, and therefore they end up looking at the same regions in visual space. And that's exactly what we're measuring here, the difference between degrees in visual space. So the emergence of overlap clustering that is purely due to these local interactions drives this contraction of receptive field, which is the main factor behind the emergence of this topography globally. But I think it's difficult to say, like, how do the local and the global interact? I mean, they interact, the local acts locally, and the only global element here is the backpropagating action potential. So the backpropagating action potential kind of allows for multiple local elements to become coordinated, right? So locally, we have multiple synapses, and through this activity-dependent plasticity rule, they become organized so that they're locally clustered. And then at the global scale, you could think of the individually locally clustered elements that then become coordinated via this backpropagating action potential. Are there other questions? So I think I want to offer you the choice. Do you want me to talk about the new topic? So do you want me to talk about what happens after eye opening in the critical period of plasticity? Or do you want me to talk about the functional implications of this clustering?
I think functional implication would be good. Yeah. That's
okay.
Me too.
Okay.
Yeah.
All right. So
well, I still want to pause a little bit. So the global, you're basically saying this global backpropagation is so I thought that this global propagation will cause like spines that are far away. They can be co-active, they be coordinated, right?
yeah.
And then somehow you will see that the receptive field difference between these spines will get smaller and smaller. Is that the right intuition?
Yeah, that's exactly what happens. So yeah, so the global signal, but again, it depends on how effective this global backpropagating action potential is. So in the case where it's very, very effective, meaning that it decays very weakly as it propagates out of the soma, then that's exactly what happens. This is the scenario here at the top, right? We get synapses far away from the soma that are still very, very closely located in visual space in comparison to the soma. So the white here, this is the receptive field offset. So it's basically the difference between where the synapse and where the soma looks at. So if the color is bright, that means that their difference in visual space is very small. So they look at basically a similar place in the visual world. So that's exactly right. So we get from a scenario where, as here, right, things are kind of all over the place. There's a mixture of bright purple and dark purple, or white and dark purple. And then we introduce this backpropagating action potential. And so what that does is it just homogenizes everything. So it effectively makes all synapses that are nearby share similar receptive field location as the soma. So if we quantify that by measuring the difference. So then the difference between the synapse and the soma becomes small, and that's the white color. So that's why we have white basically almost everywhere throughout the tree, except for very far away synapses.
Okay, there's a question on the chat room. Is there any link between the clustering structure of the synapses and the pinwheel structure for the orientation back to the visual cortex? I think it's a very good question.
Yeah, so we don't know. This is a great question. We don't know. I think the simple answer is we don't know, because the model that we're proposing is really subcellular, and the pinwheel versus this map in ferrets and macaques, in monkeys, versus the kind of salt and pepper that you see in rodents is observed at the level of kind of large scale, like networks of many neurons, so that's cellular, and this is subcellular. But we think it's the same kind of mechanisms. So the mechanism that we have here that determines the differences between the different animals is the amplification factor of the receptive fields, and because of the different sizes of the cortices. And I think it's the same phenomenon that drives, or some people would argue, forgive me now, I don't remember the author. I think it's a Korean author. I'm really sorry. He worked with Dario Ringach Pike, maybe. Sorry I can look up the reference. He had a recent paper, I forget where, Cell Reports maybe, or Scientific Reports, and he kind of argued that the same principle, namely amplification factor due to the sizes of the cortices, will be responsible also for the maps. So so it could be that it's the same principle, but the two mechanisms are unrelated, or it could be that somehow the subcellular organization then acts also at the network level to generate the maps. I honestly don't know if that's possible. I think if that were possible, it would require really a lot of mechanisms that somehow go from the subcellular to the cellular level. I'm not aware of such mechanisms, but I don't know. Anything is possible in biology.
Okay. Kai is quite happy about the answer. All right. There's another question, please.
Yeah, I think I forgot to ask this. And how do I define soma in a model then? It's like we eventually generate, for the receptive field in a center. Okay, so for example, we have a vector, right? And yeah, how do we define soma? Like, will be what's the ratio between soma and dendron?
How do we define soma? I mean, just like it's just like a synapse, so it's also described by a Gabor, but we just assume, for example, that let's see here, yeah. So we say that the soma just integrates activity from all synapses. So let's say we have a vector. So the activity of synapse 1 is x1, of synapse 2 is x2. So we label all the synapses and we put them in a vector, x1 through xn, which describe the activity patterns. And then we say that this somatic activation is just the sum of each of these activation patterns. So xi scaled by the strength of the synapse wi. So it's just w1 x1 plus w2 x2 plus so on until wn xn. That's exactly just how we do this. But yeah, we could make this, more interesting. So this is effectively a passive neuron. We don't have any active channels. We don't have anything, we don't have nonlinearities. Actually, during the review process of our paper, we were asked about adding nonlinearities or adding cooperativity. So we have a few supplementary figures if you care about making this a bit more biologically realistic. But we literally just sum up the activity scaled by the weights and then generate the signal and then threshold that. Yeah,
I think I understand now. Thank you.
Good. Okay, perfect. So I'm gonna end on just 7. All of a sudden there's more questions. More questions? Thank you. Yeah. I have to check. Yeah. Yeah, it was Dario Ringic. I think Pike is the first author. I don't know if he was the
Yeah, I
PNAS
just
author.
sent that reference in
Yeah,
the chat.
okay, perfect. Yeah, thank you for finding this. Yeah, he had
Sure.
this idea about the cortex size. Okay, perfect. Glad that you found it. So, okay, so I want to then talk a little bit about the computational implications of these things. And what I will show you just applies to the mouse. So applies to the emergence of this topography in the mouse. So this idea that synapses that are nearby will end up looking or coding for locations in visual space that are similar to the soma, and in synapses that are far away will end up looking at different positions relative to the soma. So I have no idea what the relevance of the orientation clustering in the ferret is, but I'm going to just give you a context in the mouse. And some of these ideas were already proposed in this existing paper. And so we were able to kind of verify some of them and then go beyond their findings to make some predictions. So this is actually how they did it. So they are the ones, remember, I compared this to the data, the fact that, you know, we in the model, as similar to what they find in the data, find, yeah, that these synapses nearby have a receptive field distance that's close to the soma. So the distance is increasing as you get far away from the soma. So that's the result, this topography. So now the question was, what's the implication of this topography for computations? Oops. So what they proposed to do in this experimental paper is they proposed to look at the orientation preference of the soma. This is what's shown here in the middle. And then the orientation preference of the synapse. So they distinguish between a somatic receptive field that's in the center and a synaptic receptive field that's indicated here on the side. And then, so they do this for each synapse. So they pair it, they compare it to the soma, and they divide up the space, the visual space, into two regions that they call coaxial and orthogonal space. So this is how they define them. They call coaxial space the space where within, 45-degree difference, the orientation preference of the synapse is similar to the orientation preference of the soma. And then the opposite is the orthogonal space. So for example, here, the synapse and the soma are kind of within 45 degrees similar. So this synapse falls in the coaxial space of the soma. And then this is the orthogonal space. So there might be other synapses that fall in the orthogonal space. Well, I guess they define this relative to the soma. And then they put all the synapses and they see whether the synapses lie in the coaxial or the orthogonal space of the soma. This is, for example, another dendrite. These are two receptive fields. So we see that this particular one of the synapse also falls in the coaxial space, but you see that this one falls in the orthogonal space because here the orientation preference is vertical and the orientation preference of the soma is 90 degrees. So this one falls in the orthogonal space. Is this clear? So this is a proposal that they had in this Caruso paper. They take a soma, you look at its orientation, and then you divide up the visual space into coaxial versus orthogonal space, and then you put all the synaptic inputs on these dendrites, and you look at whether they fall in coaxial or in orthogonal space. And then you can quantify basically the occurrence, so you can build a histogram as a function of the circular dispersion. So the circular dispersion is the difference in orientation between the synapse and the soma. And you can first do this for the coaxial space. So this first plot refers just to coaxial space. So we can have these, this is combination of model and data. So the data here is the circles. So maybe let's just focus on the circles because this is the experimental finding. So they bin their data into these three bins by just looking at, so here the orientation of the soma is always vertical, and they just looked at the preference of the synapses that fall in this coaxial space. So in this first bin, you put synapses that lie in the green coaxial space where the orientation difference between the soma and the synapse is 0. So you see that they're completely aligned. Both of them are vertical, the difference is 0. So you put synapses like that in this first bin. In the second bin, you put synapses that are about 45-degree difference relative to the soma. So if the soma is vertical, this is basically what you get for the synapse. It can also be in the opposite, minus 45 degree. And then for 90 degrees, you basically consider synapses that are completely orthogonally oriented to the soma. So you see that there's basically a decrease in the probability of occurrence. So that means that you see more synapses in coaxial space that are similarly oriented to the soma. We also kind of get this with the model with some discrepancy. And they proposed in this paper that the reason, so you get this kind of distribution because you get this topography. And the reason that this could be useful for computation is to encode for basically edges, so very long edges that occur in natural images. So this is, for example, a natural image, and they actually they even measured this co-occurrence of similar orientation in natural images, and they found this to match also what they find in the data. But let's just see what this means. So in this case, we assume that the soma is looking at this point in the visual image in this red square, and then they basically assume that because of this global organization of synaptic inputs, the synaptic inputs that are on the dendritic tree, on the same dendritic tree, that fall in this coaxial space will be coding for these two locations shown here in the green boxes. And they will be coding for similar orientation relative to the soma. So they found this big prevalence where the synapse and the soma share the same orientation preference. And they suggested that the reason that you have this overrepresentation of synapses that they share the same orientation preference with the soma in this coaxial space could be to encode for these elongated edges that you see in natural images. I hope that's clear. Now, they did not, unfortunately, when they looked in the opposite, the orthogonal space shown here in the purple, they did again the same kind of binning. Let's focus on the data. These are the circles. They again looked at the occurrence of a synapse that has basically the same orientation preference or slightly different or completely orthogonal orientation preference relative to the soma. They did not find the relationship in their data, but in our model, we did. So our model is here, the gray bars. So we saw that there's a slightly bigger overrepresentation of synapses that share in this orthogonal space that have an orthogonal orientation relative to the soma. And we hypothesized that this might be good for something else. We think that this might be good for the detection of collinear features in natural images. So while the overrepresentation in coaxial space tends to be good for the representation of these long edges in natural images, we think that the overrepresentation of orthogonally oriented synapses relative to the soma in this orthogonal space might be good for the detection of these collinear features. So that if the soma is looking at, for example, a region that has this horizontal orientation preference, the synapse might be looking at this one that has an orthogonal orientation preference. So you see, you could kind of detect these collinear features that exist in natural images. Now, we organized the Bernstein conference on clustering with Jan in 2019, 2 years ago, just before submitting our paper. And we invited Florencia Iacaruso to give a talk, and we showed her this, and we said, hey, look, we looked at your data, and we actually find a slightly different result from you. We do find this overrepresentation, and we like to claim that this is good for the encoding of these collinear features in natural images. And we just wondered why, or what her comment was on why they didn't see it in the data. And she actually said that they, this is a very difficult data to collect, first of all. So they originally actually saw it in the data when they were still collecting it. They had a result that was sort of increasing in the same way as we found it in the model. But then once they started collecting more data, it kind of became more noisy and more variable. And then eventually they lost the statistical significance. But she says that if, you know, she thinks this is real and that it might also be there. So potentially collecting more data might kind of further verify the existence of this bias. So we kind of leave it out there, and maybe somebody will eventually do the experiment and quantify this. So anyway, so I hope it's clear what this means by kind of dividing this space for the synapses and the soma into coaxial and orthogonal space, and looking at this overrepresentation in each of the two spaces separately. You know, we think that this organization, this global topographic organization that emerges in the mouse, could be useful for these two aspects: the encoding or the representation of these elongated edges, these collinear things in natural images, or these co-circular objects that also appear in natural images. Is this clear? Are there questions?
Yeah, it's clear.
Okay, perfect. So it's 11 o'clock, so I guess I didn't have time to show you my second part of the talk, but that's totally okay. It's actually great that people were engaged and were asking questions. So yeah, I guess we will end here. And
Yeah.
I want to ask if there's other, I mean, I can show you, I just want to show you super briefly. What I wanted to show in the second part of the talk was, as I said, to look at what happens after eye opening. There's this so-called critical period of plasticity. So eye openings around P14, P15, according to different people argue it's a different time. And in the critical period starts around 20 days after birth and ends, some people argue will never end, but yeah, maybe ends around postnatal day 35. And this is a period of visually evoked activity. Lots of cool things happen in this period. So emergence of retinotopy early on, but eventually orientation selectivity, binocularity, binocular matching, and all these other processes. And so what I wanted to show you was how circuits maintain function during this critical period of plasticity upon a particular perturbation. And the particular perturbation that was applied in this case was suture, so monocular deprivation. So experimentalists went in and sewed up the eye of the animal with needle and thread. And they did this at around postnatal day 27 to 32. And then implanted electrodes in the cortex and then investigated the in vivo activity in the cortex in the case when the eye was not closed, so normal development, versus in the case when the eye was closed. And these experiments were done in the lab of Gina Terrigiano. Part of the data was published in this paper a few years ago. And so we took the data, so spike sorted data recorded in the cortex, with and without this monocular deprivation paradigm, and then did two things. First, we analyzed the data to identify what aspects of the activity change when you close up the eye. So how does the circuit react to this perturbation of sensory experience? And then we build models to explain with sort of proposed activity-dependent mechanisms to explain what type of mechanisms can account for the changes that we identified in the data. And you know, you can, this is also published, so you can look at this in our paper from last year. So we were looking at changes in activity like firing rates and correlations, and then building network models to determine what type of activity dependent and homeostatic mechanisms can capture this in the case of this sensory perturbation. Okay.
All right.
Thanks for the attention.
It's great. Any emergent questions from the audience? No, it's very clear.
Okay, perfect. Okay, and I will be online later to answer any questions that come up during the talk, of course.
Okay, so for the second talk, you will be available, right?
Yes, I will join and I will be in the background. So whenever there's a question, we can start, we can stop and I will answer the question. Yeah.
So do you think you will have time to join the panel discussion session after your second talk?
Yes, I will be able to. I will probably have the baby with me, but because it's a panel discussion, maybe there'll be career questions about how to manage personal and professional life. So it'll be perfectly timed. So, but I will be there at 4 o'clock, right? Local time.
Yeah, local time. That's great. Let's thank Julijana again. I think it's a very nice talk.
Thanks, guys. Thanks for all the interesting questions. Very hard questions. If you want to work on any of them, and if you're a postdoc and looking to join a group, please contact me. Okay, see you later.
All right, see you. Okay, bye.
Bye-bye.
Do we need to chat?
Yeah, maybe you can chat for a few minutes.
Yeah, I think everything's going well. So.
Yeah. Julijana is still